Okay, so good afternoon, ladies and gentlemen. It's a pleasure to be here today and for the next 2 weeks. So we'll have the chance to stay together for quite a while. And well, first of all, I have to thank my sponsors and my friends and colleagues here who have organized and engineered the possibility for me to stay here for a substantial period of time, not only to give this course, but also to find out what is going on in the biotechnology, biology realm. And also, I suppose that during the course of this workshop, or whatever you name it, I would be very interested to know what you are doing. I understand that you are at different, say, levels of your academic career. Some of you are undergrads, others are masters, others are making their PhDs, perhaps there's some postdoc, I don't know. And well, I would be super interested to know about your projects, your interests, and my ambition during this course would be not to have the typical lecture course when the professor gets here and starts to deliver things and talks one after the other and you take notes and everything. So we have to make a conversation. So what I would like you to invite you from the very beginning, not to sit down and just watch to what I'm saying, but actively in what we are discussing, you know, because just keep in mind that synthetic biology, and I will try to convince you of that during the entire, say, mission here, is something that will affect you much more than me, because I'm at the end of my career, and well, you know, whatever I did right or wrong, a perspective that whatever happens in biology. So please do not hesitate to interrupt me if something is not clear. I will raise, not necessarily today, but over the period of the course, many controversial issues, because you know that synthetic biology, in a way, is looking at biological systems that leaves behind the typical, say, molecular biology. Genetic engineering is one step trying to engineer biological systems. At the end, is about engineering that issues that I'm sure that will be interesting to discuss with you, and you're very welcome. The other thing about synthetic biology, much, and I would like this group to really benefit from that, is that the idea, as crazy as it might be, is welcome. Okay, so we're in a realm, and we have this. Did you ever heard of this story about is something that you are, you know? Others. And that creates a kind of fear as intelligent. So you are under the permanent pressure and fear that your ignorance will be exposed if you perhaps stupid. In many cases, and this is typical in many workshops, any question, silence, okay? Not because you don't have questions, simply because you are afraid of being question that you are not sure whether it's good or bad, okay? So please forget that behind, and let's create an environment in which you are welcome to raise questions, even if upfront they look, you know, different or absurd or whatever. I do that all the time, and I have no problem with that. And you know, the thing is that after you get an answer to your question, you know more than before. So that's another matter. Okay, so let's try to make the most of these days that we are going to be together, and let's have fun, because the third aspect of it is that is out of fun. You can propose things, you can propose, and there's this notion that to have good ideas, you need to have many ideas. 90% of your ideas will be complete bullshit, but 1% of your ideas will be very good, but you need to have 100 ideas. Encourage this of going to examine these days, okay? With that, tell you that today we're going to have just an overview of many, many different things connected to synthetic biology. I will try to synthetic biology, where does it come from, and what were the origins, the foundational works that originated the. And I will mention on passing many topics that in subsequent say classes or in subsequent say editions or meetings will be disclosed in more detail, okay? And that's something that I hope that you benefit from. But today, do not understand some terms, you don't turn.
Sir?
Yes?
Yes. Yeah, very sorry to interrupt. Just a small note. For the online participants, there's a little bit of break in the voice, maybe a few seconds. So if some of the technical people there can take a look.
Well, I know that the speaker goes up and down and up, the technical. Is that you are biology, engineering, mathematics, and all these things, and you are not identical in these terms. That for some of you, some of the things that I will mention today will be very, very simple, and you know already, whereas for others will be completely new. So please, let's be tolerant, and if you see that I'm going to tell you things that perhaps just remember that other members of this room may not be completely aware of the jargon of biology, of molecular biology, and some words might sound a bit strange, but through the different, say, steps that we will have in this course, you will learn about everything. Okay, so this course or this workshop or this conversation is intended to be an introduction to synthetic biology. So by no means it will be an exhaustive analysis of everything that is going on in the field, because it will be impossible. But I think that it will be a good exercise. So my expectation is that at the end of this course, you will have enough knowledge, enough in understanding, not only in understanding, but also on practicing synthetic biology. So about the organization of the group, of the course, what I propose, insist that I welcome your feedback, will be the following. We have these 4 hours per day. That is quite an intensive, say, of time in the day. So what I would propose is that we spend the 2 first hours then as a kind of formal presentation in my side, and then we spend the other 2 hours by discussing assignments that I'm going to give you, okay? So these assignments will be papers that I would like you to read, and then next day, obviously not today, but next day, we will have a presentation of the different papers by some of you. We will make teams at the end or something like that. And then, well, on the basis of these papers, we can start conversations on what you see novel, what is interesting, what can be criticized about these papers. And I think that that will enter some type of, say, practical work, not only these lectures that I will tell you today. So 2 of the papers that I want you to read for tomorrow is, I will mention on passing in my presentation, but please, it would be great if by tomorrow, I don't know how busy you are this afternoon or tomorrow morning, it would be great if you could take a look to them. So, and then tomorrow for the second part of the class, I will ask some of you, well, we'll see whether we ask for volunteers or we kind of make sure that at some point each of you have the opportunity to send half of the time with these more formal lectures and the other half more in a conversation, okay, on the basis of some assignments that I will give you. Other types of assignments that I will explain you later will go as follows. You know that have been involved in this, say, activity that is called the IGEM competition. So the IGEM competition is something that started in year 2004, if I remember well, and is about having undergrad students proposing the creation of biological systems on the basis of some synthetic biology concepts and biology tools for doing something of interest. It can be creation of bacteria that promote plant growth, creation of microbes able to convert pollutants into value, or degradation of plastics, but it can be things as funny as recreating with bacteria human activities. And well, at some point I will tell you more about it. But keep in mind that some of the assignments that we have at the end of the course will have to do with the fact that you will have to propose a project, a IM-like project, in which you will be asked to, on the basis of what you have learned in previous days or in previous, classes, to propose construction of something interesting. Okay, and I'm sure that it will be a lot of fun and creative, and it will be quite interesting to have that. We have a challenge, okay, and is that my accent in English is different of your accent in English, okay? So maybe the first day you will have difficulties perhaps to understand some of the words that I will say, I don't know, but I'm sure that at the end of the course we can understand with no problem. Okay? So just get used a little bit to my accent. I'm not an English speaker, as probably or not, and English is my second or third language, and therefore we all face the same challenge. Let's try to make the things run smoothly. Okay, ladies and gentlemen, so yes, let's start today with the overview that I told you that we would do for the day. Introduction to synthetic biology. So I will start with some historical perspective. But before that, let me introduce myself, despite the fact that Hartig has presented me also. I work in this center, in the so-called the National Center of Biotechnology in Madrid, Spain. That is one of the centers of the so-called National Research Council. You are very welcome to visit. We have plenty of postdoc opportunities and so on, so check the webpage, and if you are interested to come to Spain to work not only in my laboratory but in different laboratories, I'm sure that you will find many opportunities. And this is my team, where we are composed of people from different nationalities, mostly Spanish speakers, but occasionally we have Asians, we have Germans, we have, well, as is the case today in most research countries, in research centers with a certain, say, level. So, and this is where we are. It's a nice place, and, well, if you are curious, you can go through the webpage and so on. Can we take out the banner that is in the upper side? I'm not sure how to do it.
What does this say? Hide or something like that?
No, it says you are sharing the screen. Well, it's not a tragedy. We can leave it there.
Usually, this can be
Here. No, yeah.
What does this say? Stop sharing.
Stop sharing, right?
There's something else that says.
Well, you can just
hide it or something, right?
Yeah.
So,
Just
wait.
forget it.
No. Here there's something that says hide it.
Yes, there. Ocultar. This one. Ocultar. No, that one. No. One. That one.
This one?
Yeah. Let's see. Well,
That's I think the video panel.
nothing happens. There. Yeah. Okay.
Yeah.
Perfect. Okay. Yeah. Now you can click there. No, just the X there. Yeah. Okay, here we are. All right, so sorry for these technical issues. All right, so the story I want to start talking today is the following. I don't know how many of you know these two people. Maybe none of you, right? Okay, so these two people were the inventors of genetic engineering, okay? Herbert Boyer and Stanley Cohen. And in year 73, and I will mention that on passing, were the first that were able to clone a gene. Now, cloning a gene is, nothing, absolutely nothing. Every idiot can clone a gene, right? However, I mean, they were the first that figured out the concept and the tools to take a piece of DNA from one place and put it somewhere else. So they never received a Nobel Prize, but they go into history as the ones that really did that. So this is, ladies and gentlemen, the foundational paper of genetic engineering. And if you read the title, it looks like very, very unappealing, right? It says, construction of biological functional bacterial plasmid in vitro. I mean, probably you read that in the catalog of articles in a journal, and you would just pass to the next article. Okay, but the thing is that in this article, they were able to show, using technologies that today would be completely primitive, that you can take a piece of DNA from one given origin, for a given, say, organism, extract the DNA, cut it with restriction enzymes or with other nucleases that can generate DNA segments, and then by using, I insist, tricks that today would be very, say, complicated, you can grab them and make them replicate in a different vector. So this was something that by that time, it was a complete revolution. It was the first time that DNA could be manipulated. So what is involved in genetic engineering, and most of you probably have been through this, not only conceptually, but even practically, is that you start with some chromosomal DNA. Do I have a pointer here?
The main button is a pointer.
Oh, yeah. Right. Well, it's not super strong, but you can see. Okay, you have a DNA of any origin, and then you have a gene X, and then you manage to get sticky ends at the end of a piece of DNA that has been cleaved previously. These sticky ends at some point could be generated through restriction enzymes. Before restriction enzymes, it was possible to make this type of operations with a more complicated technique, but a big difference was made by the discovery of restriction enzymes. And as you may recall, restriction enzymes leave, say, protruding ends that now recognize equivalent bases in the other, say, recipient DNA here. So you have a vector, for instance, a plasmid, a circular DNA, and you digest with a resistant enzyme. You take DNA with the same and treat it with the same enzyme. Then you can put them together, and here comes one of the most important enzymes of the history of genetic engineering, and it's the one called ligase. So because ligase is able to take the gene that you have excised from somewhere else and ligate it, as its name indicates, to the vector, to the plasmid here. And at the end, you have a so-called recombinant plasmid. And then this recombinant plasmid, by that time, could be introduced in bacteria. Now recombinant constructs can be put all over the place, in plants, in mammals, you name it. But by that time, only bacteria were able to receive these constructs, and in many cases, this gene that came from somewhere could be produced in bacteria. Okay, so it was a complete revolution. And we're still living out of that revolution. Okay, so the second, say, big thing in the story is that the technology developed to the point that you could clone not only, say, constructs and DNA fragments and plasmids and so, but even live systems. Because if you take DNA from one place and put it into the other, you generate a manipulated living being, right? And by that time, we are talking year 72, there was this big discussion that you are welcome to have an opinion on that, about patenting life systems, okay? So far, you could patent, I don't know, cars or pieces or, but this gentleman here, Ananda Chakrabarty from Kolkata, but he was a scientist in the United States for many years, was the first that filed in year 72 a patent case on a bacterium able to degrade petroleum. So he was using by that time a technology that is called plasmid breeding, a kind of primitive type of genetic engineering. And then he started his patent in year 72, and it took nearly 10 years to have the outcome of this legal case approved as a patent, okay? So the bacterium that he engineered was, well, he's sitting here happily in front of the Supreme Court of the United States, and the bacterium itself did that. You started with a culture with petroleum, and then after application of the petroleum or much of the petroleum was degraded. And well, I have to say that the bacterium itself was not a big deal. It never made it into a commercial application or so. But conceptually, it was a big breakthrough because in the history of biotechnology, because it was the first time that a living entity manipulated by humans was subject of a patent. And this is, well, by the way, this is myself with the same guy many, many years later in one meeting that at some point I participated. Obviously, we were much younger by that time. Okay, so cloning recombinant bacteria it was the basis of many, many biotechnological developments. And I want to mention like three big milestones in the history of biotechnology. One of them was this paper that I just mentioned in year 73. That was the construction of this bacterial plasmid in vitro that I just mentioned, 73. Now, years later, there was a super big hit in the field of biotechnology. That was the cloning and expression of the human erythropoietin gene. Erythropoietin is a hormone that is super essential for regeneration of blood, of red blood cells. And I don't know, you have some members of your family going through a kidney dialysis, but before this enzyme or this hormone was available, people subject to recurrent dialysis live very shortly because the procedure eliminated a lot of red blood cells, and therefore, the production of erythropoietin was super difficult. But by cloning the gene of erythropoietin that comes obviously from a mammal source and expressing that in a bacterial system, then now erythropoietin was not an issue any longer, and that helped tremendously the survival of patients of liver diseases. So it was a super big thing, and you know, out of money, companies, you name it. And then many years later, like 20 years later, there's this paper that I consider that the ultimate, say, production or say deliverable of what you may call traditional genetic engineering. And it's this paper here, 2006. There is the production of artemisinic acid engineered yeast. Well, you know about malaria, and you know that malaria is very difficult to treat, but one of the say, products or chemicals or medicaments that can treat malaria is artemisinin. So artemisinin is produced by a plant that is cultivated in Asia in large extensions, but require obviously to have a crop, to extract the plant biomass and so on, and at the end produce the artemisinin. And it's a very complicated process. So the laboratory of someone called J. Kisling, and this is a name that will pop up various times during the whole course here because it's a kind of big name in synthetic biology, published this paper, a landmark paper, in which they were able to engineer production of a precursor of artemisinin in yeast. Obviously, yeast is not a bacteria; it's a bit more complicated. We will return to that much later. But indeed, it was one of the cases where, by having a rational isolation of the genes involved in artemisinin production, put them in yeast and making some tweaking, then they were able to produce this important compound in a bioreactor. Okay, and we are here. We are getting to the end of the history of what you may call traditional genetic engineering. Let me tell you on passing, and this is something that we will discuss in one of the talks of this course, that what do you think? You think that producing in a fermenter large amounts of this compound is a good thing or a bad thing? Who thinks it's a good thing? Raise your hand. Yeah, don't be afraid. Okay. Yeah, good. Who thinks it's a bad thing? Okay, the rest don't have an opinion? Okay, so the way you say, well, we have this compound, it's super difficult to produce, we have to go plant it and everything, and now we have this wonderful thing that in a reactor of 20 liters, we can produce as much artemisinin as in various hectares of this plant. Do you think it's a good thing or a bad thing? Well, in principle, I mean, intuitively you say, well, this is great, this is a fantastic thing, right? Because you can make now the drug cheaper and everything. However, there is a consequence of that. and it's that all farmers that made a living out of artemisinin production went out of job, right? So this is, I want to raise this very early in the whole discussion because, don't think that synthetic biology is completely free of consequences. Everything that we will discuss in this course will have some type of societal repercussions because the technology is so powerful that it's able not only to change production of this or that, but really have a tremendous impact in big sectors of our economy and our society, okay? So in these IEM projects that I mentioned before, there is always this kind of separate study on societal consequences of any development. For instance, if you propose, let's make a bacterium able to eat plastic, for instance. Okay, you say, well, this would be wonderful. Okay, let's think it twice. Maybe we'll have to think it twice. No, I would not give you a solution for that because it's a bit complicated, but I leave you with that. So some propositions that we make from the point of view of synthetic biology. First, in a first impression, they are wonderful, they are nice, but sometimes there are these secondary consequences that may not be as good. Well, in any case, so we get with these stories to the end of genetic engineering. So you make this big jump in time, and then we come here, and we come to the times of systems biology. So systems biology and the massive application of technologies for identification of genes, identification of transcripts, identification of interaction between them and everything, lead us to this case, and this is what some scientists call the hairy monster. So you study a system, you can identify how many genes are involved in a given process or in a given organism. You can determine through a large variety of technologies, omics, genomics, proteomics, metabolomics, interactomics, you name it, at various scales, at various whatever. So you can make this type of maps where you have a kind of interpretation, a kind of visual depiction of what is going on in a life system, okay? But the question is to make sense out of that. So that has created, and I have to say that synthetic biology is something that comes as an immediate consequence of systems biology. We face a new situation, and this new situation is the fact that we have many different, a lot of data from biological systems, and we are facing unknown objects and unknown, biological, phenomena. So when you face something new here, what questions, you walk out the street and you find this, what would be your first thought, okay? Okay, so there are various questions that you can think. First of all, where does it come from? Is it extraterrestrial, or is something made by my neighbor? Second question, very important, how does it work? Is it useful for something? And third, connected to that, can we in fact use it for something useful? So you may say, well, this kind of flying saucer, I don't care where it comes from, but maybe it has metal, and I can take the metal and sell it to someone, So don't think that the questions are always the same, they can be different. So the same happens in biology, okay? So you can raise the question where things come from. This is the typical question of evolutionary biology. How does it work? It's a completely different question. And one of the punchlines that I would like to convince you today is that the questions in biology have to be really well and precisely formulated, because if not, we confuse terms and labels, and then we get very messed up. So one question is to ask where things come from, and a different question is to ask how do they work. And of course, a different question is, once we know these two, can we use it for something useful? Okay, so you say, well, where do biological entities come from? That question is kind of solved, more or less solved, okay? And there are two pillars in biology, as you may know. That is evolutionary theory, Darwin and some of his, colleagues there during this period of time. And then DNA structure and so on. So between these two, contributions, we can understand conceptually, and maybe not in detail, but basically we can understand where biological systems come from. Okay, but there's another interesting question here. As I said, a different question is to understand how they work, where they come from and how they work. So in molecular biology, the kind of traditional view is one concept that was elaborated time ago by this gentleman called Francois Jacob, that is one of the fathers of synthetic biology, working for a long time in the Pasteur Institute. By the way, I had the pleasure to meet him in the year 2008. He was still alive in the price that Kartik mentioned before. Okay, so he developed this idea that he called tinkering. So tinkering is equivalent to the word in French called bricolage, and sometimes, what happens in biology, and I don't know whether you have noticed that, is that the terminology in many cases is very ambiguous, okay? So if you say the distance between here and here is 50 centimeters, you know exactly what you're talking about. If you say biological systems evolve by tinkering, so tinkering is a very vague concept. So it gives you an intuitive idea what is going on, but sometimes you don't understand. Or fitness, in evolutionary biology, what is fitness? So these are vague terms. And one of the missions of synthetic biology, as a matter of fact, and I will return to that later in a subsequent class, is to really make standards, not only standards for measurement, not only standards for assembling genes, but also semantic standards. Semantic standards means that when you say one word, you have an unequivocal meaning for that word. We will return to that later. Well, in any case, so in every molecular biology book, you will find this idea of tinkering. So tinkering is this concept that, well, in evolution what happens is that you have many things around, and in a very casual manner, two of these two things can get together, and maybe they improve a little bit the functionality in respect to what you had before, and therefore through Darwinian, say, selection, this combination is selected, and then you move on, and then you move, and then you move off, and you move on. So the idea here is that there's no, any specific reason for things to get together, but things get together in a kind of hazardous, a kind of random procedure, and then if they hit a point where they do something useful, then evolution takes care of it and will move the system to a next step.